Hello and welcome back to Kim Reads as we continue with Rosalind Franklin. Chapter 5, A Crucial Discovery. You might not realize that we already know a lot about DNA. You know that traits like the eye color or height can be inherited. You know that tomato seeds will always grow into tomato plants, which will in turn produce other tomatoes with their own seeds. Tomato seeds will never grow corn. In the same way, dogs will always have puppies, not kittens. And the puppies will resemble their parents. You know these things without thinking about them very hard, but they're true because of a substance called DNA. DNA stands for, bear with me, deoxyribonucleic acid. You can see why it's just called DNA. Every cell, plant or animal or human, contains DNA. Usually tightly coiled into chromosomes, which come in pairs. An offspring gets one of each pair of chromosomes from each parent. Humans usually have 23 pairs of chromosomes, for a total of 46. Mice have 20 pairs. Tomatoes have 12. DNA is the blueprint of every living thing. But in 1950, no one really understood what it was. Scientists knew it was an enormous molecule. They knew it consisted of these three parts: phosphate groups, sugars, and nitrogen bases. There were 4 different types of nitrogen bases called adenine, guanine, cytosine, and thymine. The amount of adenine in a segment of DNA always equaled the amount of thymine. The amount of cytosine always equal the amounts of guanine. But scientists didn't know how the 3 parts were put together. Understanding the structure of DNA would unlock an entirely new branch of biology. It would change the world, and science knew it. All across the globe, researchers were studying DNA. Rosalind Franklin came to King's College London in January 1951 to work in the X-ray diffraction laboratory run by a man named John Randall. Another man there was named Maurice Wilkin, was already working on the DNA with old, outdated equipment. Randall let Rosalind install newer equipment. She spent the first 8 months just setting it up. Rosalind was friendly with everyone in the lab, except Maurice Wilkins. They disliked each other right away. Wilkins was quiet and shy. He didn't like loud people, and Rosalind was certainly loud. He'd also been struggling to discover a way to keep his DNA samples in a form that made them easier to photograph. Rosalind not only told him how to do it, she told him the technique had been discovered a long time ago. Maurice was embarrassed. They quit disliking each other. They despised each other. Meanwhile, back at Cambridge, 2 scientists named James Watson and Francis Crick were also working on the structure of DNA. And so was a famous scientist in America named Linus Pauling. In November of 1951, 11 months after she returned to London, Rosalind gave a lecture about her research. James Watson traveled from Cambridge to hear it. Rosalind showed early photographs of the X-ray diffraction of DNA and concluded that DNA formed helices, that is, it spiraled around itself, with the phosphate groups on the outside. James didn't take notes or listen very carefully, nor did he believe Rosalind's theory. A few months later, he and Francis proposed a DNA structure that had a phosphate group on the inside. Linus Pauling thought they were on the inside of a molecule too. It was tricky and complicated work. The math needed to interpret the photograph was difficult and took a long time. Plus, it turned out the DNA could take 2 different shapes. Rosalind called them A form and the B form. This meant that the results from one photograph didn't always match the results from another. Rosalind persisted. In May 1952, she and her research assistant, Raymond Gosling, were able to take a beautifully clear X-ray photograph of the B form of DNA. It was the best of their many attempts. They labeled it Photo 51. It doesn't look like much to us, but Rosalind proved DNA structure was not simply a helix, but a double helix, two spirals joined in the middle by the nucleotide bases. Rosalind continued working on mathematical proof. She also started working for a different job. She didn't want to work with Maurice Wilkins anymore. John Randall, Rosalind's boss, told the researchers at Cambridge about Rosalind's work. On January 30th, 1953, James Watson came to Cambridge to talk about it. He and Rosalind ended up having an argument about DNA. James thought Rosalind's structure was wrong. Maurice Wilkins, though he still hated Rosalind, knew she was right. As James Watson was leaving, Maurice Wilkins secretly showed him photo 51. When James saw it, he later wrote, "My mouth fell open and my pulse began to race." But to really understand the photo, James also needed Rosalind's calculations, which he was able to get and read. Maurice never told Rosalind he shared her photograph. He didn't ask her permission to share it. A few weeks later, Rosalind took a job at Burbank College. She had to leave all her DNA research and many of her notes at King's College. They belonged to the laboratory, not to her. But before she left, she wrote the scientific paper about her discovery and sent it to be published. Scientists use scientific papers to share knowledge. They also do it to claim ownership of their discoveries. The first person to publish a discovery gets the credit for it. James Watson and Francis Crick beat Rosalind to it. As soon as they saw Rosalind's photograph, they changed their theory of DNA structure. Like her, they now believed it to be a double helix. They never gave Rosalind the credit for her work or admitted how much they learned from it. James and Francis' paper on the structure of DNA was published in April 1953. Rosalind's papers, she wrote more than one, were published afterward, and they were seen as confirming James and Francis' ideas. The truth was the other way around. And that's the end of that chapter.